So welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effects seminar series. My name is Diego, and I'm a member of the VES organizing committee, along with Stephen Airwood, Mireia Seoma, Jan Ilbudu, and Adrini D'Souza. These seminars are made possible with support and a lot of help from Laura Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas Variant Effects Alliance. So a huge thank you to them. As a reminder, we are always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I have some housekeeping items to mention. Each talk is about 20 to 25 minutes, but there should be time for some questions. Please post any questions you have at any time to the Zoom Q&A, and we'll read them out to the speaker at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can see the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. You don't have to worry about copying the URL. I'm going to post all these links to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. If you've enjoyed these seminars, then be sure to register for the Mutational Scanning Symposium, which is going to be held online and in person at the Wellcome Genome Campus in the UK on July 13th and 14th. The website listed here has all the details. Just keep in mind that abstracts are due by the 16th of May. And for the last housekeeping item, this is rarely an issue, but just as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So without further ado, we have two excellent speakers scheduled for today's presentation. First up, Dr. Samuele Cancelieri is a postdoctoral fellow at University of Trento. His work focuses on software and algorithm development for CRISPR target prediction using single nucleotide variants and indels, and also analyses for finding gene isoforms related to neuropsychiatric diseases. The title of his talk today is Human Genetic Diversity Alters Off-Target Outcomes of Therapeutic Gene Editing. So Samuele, go ahead and share your screen and start whenever you are ready.
Can you hear me? Okay, so thank you. First of all, thank you for inviting me, and welcome all to this presentation. I am Samuele Cancelieri, and topic of my presentation, as already said by Diego, would be how the genetic diversity, so variants and indels, alters the outcome of target prediction and also of target results when we use CRISPR, for therapeutic gene editing. First of all, I want to briefly describe what is CRISPR for anyone that doesn't know. CRISPR is a mechanism used by bacteria mainly, and basically is a sort of defense against viral infection. When this mechanism was detected and discovered, we tried to engineerize this system, this mechanism, to try to modify DNA sequence in cells and in vivo targets. But why is important to understand and validate CRISPR result and CRISPR? That's because the mechanism is used by bacteria, and so is not very precise, very effective when we try to use it on human cells, for example. That's because the mechanism uses an RNA sequence, basically complementary to the DNA sequence we want to modify. And it's possible that the complementary sequence on the cell, the DNA that we want to modify on the cell, is not completely identical to the RNA sequence, it's not completely complementary. And so we will have some problem known as bulges and mismatches. And so it's important to understand where we are going to cut, we are going to modify the DNA since we want to treat humans. Also, to keep in mind is that each human is genetically diverse in its the DNA sequence, and so variants like SNPs and indels are really important, and to take into consideration because, for example, a sequence that is can be therapeutic for a specific person can be problematic for another one, just like you can have some reaction to a specific drug. So we basically understand that this is a big problem when you want to treat humans. And so we try to understand how the variants can affect our result and possibly impact the outcomes of the treatment. There are different kinds of impacts. For example, the variants can impact different functional annotation. So for example, variants can be in an exon or in a promoter or in answer in a specific gene. And if we don't account for that, we can modify a potential dangerous target in a person with that share, for example, a SNP in a position. Or we can, for example, have the risk to target to produce off targets also in a population, because variants are moving like populations, and so different population can have different variants and different indels. Also, we have to consider that even if variants are moving like a population, a single person can have different variants and response in a different way to treatment with the CRISPR drug. And so we concentrated more in the final part, so population and individual level understanding of how variants are affecting CRISPR. We have a real case study. Here I will present a real case study, and we started by targeting the BCL11A gene with SPCAS9 protein, and we tried to predict of targets using the 1000 genome PCR data, so basically the variants extracted from the 1000 genome. SNPs data and also indel data, single nucleotide variant and indels. We started by taking the reference genome, the HG38 genome. We applied the genetic variants, and then we use the sequence that I will show later. We use a sequence that is developed to target the BCL11A BCL11A gene. And then after we try to predict the outcomes, we try also to annotate to understand where possible targets are on the human genome and in the specific population included in the 1000 genome project. This is a brief summary with some results. As you can see, the introduction of variants generate a lot more new targets. For example, if we check targets with 3 mismatches and 0 bulges, we have 12 more targets that can be potentially problematic. To better understand this, we generated this score distribution plot, and in this plot you can see that the off-target we will talk today is generated by the introduction of a single nucleotide variant. And the score that we calculated, known as the CFD score, a well-known score in the CRISPR sector used to understand the cutting power of the target, so the probability that the target will be cut in the resulting genome. These SNPs introduce an off-target that basically have 90 point more score result, from 0.02 in the reference sequence to 0.94 in the variant sequence. That's because the SNPs changes a C to a G in the PAM sequence of the guide, and basically allows the cut in an intron in the CPS1 gene. This variant is commonly shared in the African and African American population. This is the minor allele frequency taken from the GNOME-AD project, and can result in the inversion due to the cutting in this specific position, and the inversion of the chromosome. This is another test we have done, including also the HGDP variants in the population. And as you can see, we find a lot more candidate of targets with an increasing in the CFD score. The red dots are targets with the reference or 1000 genome variant, and the blue dot is are targets, sorry, when we include also HTTP variant in our genome. Also, we try to understand how this can affect different population. And so we generate also a distribution plot that basically shows how the introduction of more variants, in this case with the HGDP project variant, we find new possible targets, new candidate of targets, and how they are distributed with different population. And for example, in the African population, there are we need only few individuals to generate a lot of more of targets. Also, in this with this plot, we wanted to show how these targets can be shared by the same population. So, for example, we have 60 new targets in the African population, but more than the half of the targets are private, so are shared by only one individual in the population. So also with very rare variants, it's possible to generate problematic of targets. This is an extract from an individual of the East Asian population in the HGDP variant dataset. And we found that this individual have a very rare of target that with a map of 0.0005, so very, very rare, that can lead to a problematic of target when treated, for example, with this sequence. This is the original sequence that we used to treat the BCL11A gene. This is other kind of distribution that we generated starting from the common distribution with all the population. We basically extracted all the different population, and we analyzed how the distribution of targets changes when we try to when we use more individual from the same population. And as you can see, when we use individual from the African population, there is an explosion in the number of targets. With other population that shares less variants, we generate a less more targets. And then this is a more specific analysis on how the SNP that I talked before, so the SNP that generated a very dangerous of target in the that is shared in the African and African American population. How after the treatment the result alignment of the treated cells changes when we introduce the SNPs. Basically, this is extracted from a cell that comes from an individual that have the SNP, and how the SNPs is affecting the result of the possible cleavage and the amplification. And how you can see, when we try and we test the sequence in the cells extracted from the donor with the variant, we have some cutting on the DNA of the cell, and this is not a great percentage of cutting, but it's can be problematic and has to be taken into account. This cutting can lead to an inversion when happened on the DNA, and so, as I said before, even if it's very rare, we cannot avoid to understand that this can be problematic. And this is some plot to better see how the alternative sequence can lead to the inversion that they took. This is other amplification that we generate with the donor sequence. Okay. Also, we try to use different guides, because the guide we tested is specifically designed to target the BCL11A gene, and to see if the problem can be just to use a specific RNA sequence that maybe has a lot of complementary sequence in the DNA, or the problem is common and can be seen when using different guides. And in this heat map, we are showing that when testing with other guides, we have a lot of targets, that are generated even with very rare variants, and also that few targets can have a score really high. A really high score means that it's very probable that the sequence will lead to a problematic cut in the DNA sequence for example, in this case, in the human genome. And this is a plot that just to show that a lot of the possible targets can be in problematic location in the genome. For example, on tumor suppressor genes, or in coding sequence genes, or for example in PAM creation position, so in position that includes a new PAM sequence to generate a new PAM sequence on the host genome, and that can be very problematic, because, for example, if we are not targeting a tumor suppressor gene and we modify the sequence of the gene, that can be dangerous. That can happen a dangerous outcome, and maybe we can that can result in a possible disease for the people treated. Those are plots that we generated, like the first one I showed with red and blue dots, and they are generated to show the distribution of possible problematic targets on the genome, and how variants are affecting the result. For example, with pancreation, that is one of the most problematic outcomes we have when introducing variant in the human genome. We can see that there is a lot of increase in the CFD score, big increase, in the CFD score, because PAM sequence is basically one of the most important complementary sequence when we are using CRISPR. Also, these are other predicted targets with new guides. For example, this guide that is targeting EMX1 gene with an alternative variant, MAF 0.52, so very common. We found that there is this possible cut on the HSF1 intron. Or for example, in this case, we are targeting with this sequence the PCSK9 gene, and the introduction of this very rare variant, we have this possible off target. And so what I want to say with this presentation and with this information is that variants are very difficult to treat and very difficult to understand when you are using CRISPR-Cas and other type of gene editing mechanism. And so the software we developed and the presentation I made was made with the aim of just to say that it's very difficult to treat and to understand this kind of variant. We made a lot of work to create this software and this presentation. And when you're working with gene editing and with this complex problem, it's very difficult to understand and to predict the outcomes. Those are many outcomes that were predicted by software. The wet lab part was very complex and very problematic, and you have to pay a lot of attention when working with this kind of data. And the software that I will briefly present the page and how you can download it. And the software is I hope helping to generate data that are trustful and can be used to maybe produce some useful treatment. This is the full page. The first one is a web page you can visit, is open to use, and you can use different genomes, you can use different RNA targets if you have it. And if you want to work offline, we have a Bioconda package that is available here. And also this is the page in the Pinello Lab GitHub repository, where you can download the software and also use the Docker repository. Thank you very much for your attention. And if you have any question, I'm here to answer. Thank you.
Thank you for your fascinating presentation. It was really good. While we are waiting for people to type their questions in the Q&A session, we have some questions for you. So the first one was submitted by Diego, and he says, let's say I'm trying to edit a genome, but I want to minimize the possibility of off-target effects. How would you recommend that he goes about that? Would they have to sequence the target genome to ensure there's a low chance of target?
I'm not sure I completely understand the question. You were saying that if you want to target a specific position in the genome, how you have to select the target? Is that the...
Yeah, to reduce possibility of target effects, would you have to sequence your target genome to ensure that there's a low chance of the off-target effects?
Yeah, I think yes, that's correct. In this case, I'm not the one that selected this sequence, because the sequence that I show, for example, is the sequence that generates this result is not chosen by me. Was chosen by very long testing, but usually you select a specific region that you want to treat, And then you try to generate as much as possible complementary sequence with an RNA sequence to the DNA you want to target. And then you try to predict all the possible outcomes, for example, using this software. And obviously, the last thing you have to do is our experiment, wet lab experiment, to better understand how the outcomes.
Okay, so I actually have a question. It's about the reference genome that is used for the alignment, because we have an older version of the reference genome that is GRCH38, but there was a newer version that has the complete sequence that were lost in the first genome, that is the CHM13. Would using one versus the other would impact in the offer target prediction?
Yeah, for sure. If you change the reference genome, you will have different results. Very conservative regions are obviously maintained when they update the reference sequence. But yes, if the reference sequence is changing a lot, also the outcomes in the predicted targets will change. Yes.
And which one of them do you use? And I actually have a follow-up question for this. What do you think would be a valuable strategy to have reference genome, their population is specific, let's say African reference genome complete sequence. So the target prediction is target for that population and the genotype from that diversity background?
Yes. What we have done is very similar, because we take all the variants included in the 1000 genome project, and we take all the variants and we generate a reference genome containing all the same variant at the same time. And then after the alignment phase, we extracted each of target, and we basically assign each of target to a population, even more population if a variant is included in more than one population. So yes, generating, obviously the aim is to have a perfect genome for each population that you can use as a reference. That will be wonderful, but I don't think that will be very feasible, not in a few years. And so yes, but obviously that's the best you can do. Generate the reference genome for each population would be the best possible
Perfect.
reference genome to use. Thanks.
Let's see if someone else submitted a question. I don't think so. If you want to submit your question, we still have a little bit of time. So I have one extra question for you, if that's okay. Do you see a correlation or an enrichment of the type of mutation that you see more for the off-target effects? As an example, you saw a transversion for C to G. Do you see more transversions or more transition, or is like random?
I'm not sure, because I'm not a genetist, so I cannot answer. I don't think I can answer correctly this question. I think that the likelihood to have transversion SNPs is very common. So yes, I think this is the most seen single nucleotide variant that you can find. This is just my answer. I don't have the data to support that. I don't have done this research on our result, but yes, I think that's very likely to happen.
Thank you so much for your presentation. It's a very important research, and we can continue this conversation in the AVE Slack channel.